Hey everybody, welcome to the Enzyme Lecture. So we're going to talk about enzymes and what enzymes are. And enzymes are actually really cool, and they're crucial to survival. Without enzymes, we really couldn't do much at all. So enzymes are really important to all living things. So what are enzymes? So enzymes are proteins, either tertiary or quaternary structures that catalyze reactions or accelerate reactions, chemical reactions in our body or in living things. And another really important characteristic about enzymes is they're not changed at all in the process. So they are kind of, they come in, they speed up the reaction, and then they stay unchanged. They can keep being used, which is really important concept because in a human body, we don't need to worry about continue to make more and more enzymes. We can, once we make the sufficient amount of enzymes, we're good. We don't need to make any more. So that's really important because that saves on a lot of energy. So a few important points there. Again, here's a model of an enzyme. You can see here, we're going to talk about this and what that is in that space. But again, important things to know, enzymes are proteins. Definitely want to know that. The word that describes enzymes is called a catalyst, which means it accelerates or speeds up chemical reactions in the body. And it's also important to know that they're not changed in the process. So enzymes, they are very specific to what they will catalyze. So they will only catalyze one single thing. And so they're very specialized, which is important because we don't want enzymes to be catalyzing all sorts of different reactions, which would really create chaos in our body. So it's great that enzymes only will catalyze the specific reaction. So it's important to know that. Like I said before, they are reusable. And another important point in science, a lot of times the words end with the same suffix. Like in enzymes, most enzymes end in ase, like lactase, galactase. So those catalase, those words kind of give away what it is, the function of the word, or what the function of the subject. So if it's anything that ends in an ase, A-S-E, it's usually an enzyme. So that's an important point. So sucrase, lactase, maltase. So again, sucrase, what do you think that goes ahead and catalyzes? The making of sucrose. What does lactase catalyze? The production of lactase. What is maltase? The production of maltose. So you can kind of give an idea here. So here's the structure of an enzyme. This is the enzyme itself. The green thing is the enzyme. The thing that the enzyme acts upon. So in this case, in sucrase's case, it will act on sucrose. Lactase's case, it will act on lactase. So whatever the object, the molecule that the enzyme will act upon, is called the substrate. So that's very important. And then where the substrate fits into on the enzyme is called the active site. So both of these are very important. You'll definitely see those on the test. So how do they exactly do this whole catalyst thing? How do they speed up these chemical reactions? What exactly allows them to do this? So this is kind of a short answer type question because it's that concept. You have to be able to explain the concept. So enzymes, the way they work is they weaken bonds in the molecule. So if we're talking about sucrose and sucrase, sucrase will lower or weaken the bonds in sucrose, which makes it much easier for the reaction to occur. And the proper term for that is it lowers its activation energy. So if you look over here at the chart, every reaction that takes place, every single chemical reaction that takes place usually needs an initial input of energy to get it going. So you see here, here's our reactant, So you need a certain input of energy, so total energy, input energy, and then the reaction will occur. So there's always some input of energy that is needed. And what enzymes do is they lower this energy. So before it might have been up here, but now with the enzyme you only need a little bit of energy, which is great. It's a great thing for living things. Living things hate to waste energy. So anytime we can save energy, it's a great thing. So here's without enzyme. So you see here the amount of free energy. It takes a lot of energy for it to, a lot of input of energy for the reaction to occur. But with an enzyme, it's lower. Therefore, it makes it much, much easier for the reaction to occur. Not as much energy is needed to be put into this at the start. We'll be dealing with your virtual lab on Monday. We'll be dealing with these types of reactants. So the enzyme without substrate has no reaction with hydrogen peroxide. And so, sorry, let me break this down. So the enzyme we're using is called catalase. In this example, catalase enzyme. And what it does is it takes hydrogen peroxide, which is H2O2. You usually use that on on cuts and to clean cuts and stuff like that. So what it does is this enzyme catalase will break H2O2, hydrogen peroxide, down to water and oxygen. And that's usually why you see bubbles when you put hydrogen peroxide on a cut. That's because it's using catalases in your body, and it breaks the hydrogen peroxide down to water and O2 bubbles, and that's why you get bubbles. So without the substrate, you don't really see anything, but with the substrate, you see a bunch of bubbles. So again, we'll take a look at this more when I get back. The enzyme substrate, let me just move that. The enzyme substrate complex. So this is when I go back to, so you're going to probably get a picture similar to this where you have to label. It's pretty easy. Enzyme is pretty recognizable because it usually has this nice part that the substrate fits into. So what you're going to probably need to do is you're going to need to, given this picture, you're going to need to tell me, oh, this is an enzyme, oh, this is the substrate. And then also, you're going to have to label this as what's called the active site, where the substrate joins. So again, just to remind you, the substrate is what the enzyme is acting on. So the active site, like I spoke before about, is the restricted region of the enzyme molecule which binds the substrate. So where the substrate binds to on the enzyme is called the active site. And I call it a restricted region because it's very specific. This is where the specificity comes in. It has to almost perfectly fit into the active site. The substrate has to almost perfectly fit into the substrate. Substrate has to perfectly fit into the active site for it to work. If it doesn't fit, it won't work. So there we go. So that idea of that perfect fit, we call that induced fit. It's when the substrate starts to bind to the active site, it's going to kind of push its way in and kind of induce its fit and kind of form that perfect fit in the enzyme. So you see this here. Let's see if we can, here we go. So you can see kind of almost it's a perfect fit, kind of squeezes its way in there, enzyme acts upon it and breaks it apart. So that's a perfect example of how an enzyme works. Pretty cool. So again, the induced fit is a change in the configuration of enzyme's active site. So it changes a little bit for the substrate to fit in perfectly. And again, it's induced by the substrate. There you go. And then finally, what affects enzyme activity? So what kind of things affect enzyme activity? You may have heard that you could potentially die from a fever. And the reason you could potentially die of a fever is because temperature can do what's called denature the enzyme. making it non-functional. If your enzymes don't function in your body, you will die very quickly. So we're first going to look at temperature. Is the most dangerous. That can completely denature, is the proper word, denature the enzyme completely. And then pH can also denature the enzyme, making it non-functional. And in addition, sometimes salt concentrations will also denature the enzyme. And then I talked about cofactors, and the next thing is cofactors, coenzymes. So if you ever wondered what vitamins do for your body, obviously it helps you keep healthy, right? Well, specifically, what does it do? This is what vitamins do. Vitamins will help enzymes function. So cofactors are inorganic substances, which means they don't contain carbon at all. Like zinc and iron are two primary examples of cofactors. And then coenzymes are like vitamins. They have carbon in it. And so both of these, what they do is they help the enzyme work properly. So an example is iron must be present in hemoglobin in order for the blood cell, which contains hemoglobin, to pick up oxygen. And then vitamins, of course, when you take in vitamins, they help your enzymes function. And when your enzymes function really well, you feel good. You feel healthy. You're able to function overall very, very well. So that's what vitamins do pretty much, is they help enzymes in your body function. And that's it, folks. Thanks. I'll see you on Tuesday. Bye.